Welcome to Hands-On Biology. Today, we are going to discuss inheritance, Mendelian genetics. Genetics is the study of genes and how different characteristics of an organism are transmitted from one generation to another. It can be defined as the study of heredity. The study of genetics was started by Gregor Mendel. He carried out several experiments on garden peas. Gene. This is the basic unit of inheritance. It determines the characteristics of living organisms. For example, physical characteristics like height, color, size, weight, and sex. Allele. This is an alternative form of a gene. According to Mendel, there are two alleles for each character, for example. If it's height, there is tallness or shortness. Color. It can be either black or white. Sex. Female or male. These are basic examples. Mendel carried out many crossings to study the inheritance of characters. Using height, he crossed pure-breeding tall plants with pure-breeding short plants. He obtained only tall plants. He called this the first filial generation of offspring. He wasn't able to obtain the short character. He then crossbred the plants from the F1 generation. These are tall plants. He then obtained both tall and short plants. This means that the short character was masked in the F1 generation, but expressed itself in the F2 generation. Such characters are called recessive characters. They are determined by recessive genes or alleles. The strong characters that mask recessive characters are called dominant characters determined by dominant genes or alleles. Genetic crossing.
Welcome to Hands-On Biology, Competency-Based Learning. Struggling with biology? Not anymore. Here, we break down complex biology concepts into simple, clear, and engaging lessons. Hit that subscribe button now and take your learning to the next level.
This is the representation of the transmission of characters. Letters are used to represent genes or alleles. The capital letter is for dominant, and the small letter is for recessive. Let capital T represent the allele for tallness, and let small t represent the allele for shortness. Parental phenotypes, tall plants crossed with short plants. Parental genotypes, pure breeding, capital T, T, with pure breeding, small t, T. According to Mendel, two alleles for each character. Meiosis leads to the formation of gametes. Alleles separate during gamete formation. This is followed by random fertilization. Each allele has equal combing with either of the alleles from the other gametes. This forms the F1 generation genotype, or the genetic composition. In the physical appearance, all plants are tall, crossing plants from F1 generation. Parental phenotypes, tall plants crossed with tall plants. Parental genotypes are heterozygotes, and have both capital T and small t. Meiosis leading to the formation of gametes. Alleles separate and form gametes, with each allele in the different gamete. This forms the first Mendel's law of genetics. We are going to dive into its details in the new A-level adapted competency curriculum. The characteristics of a diploid organism are controlled by alleles occurring in pairs. Of the pair of alleles, only one can be carried in a single gamete. This is also known as the principle of segregation, random fertilization to form the offspring genotype of the F2 generation. In physical appearance, 3 are tall plants and 1 is a short plant. Diving into some terms. Gene, a segment of DNA that codes for particular characteristics. Examples of characters are height, weight, and sex. Allele, a variant or alternative form of a gene that exists at a specific locus on a chromosome. For example, height has 2 alleles, for tallness and shortness. Capital T, this is tall. Capital T with small t, this is also tall in physical appearance. The capital T, dominant, masks the small t, recessive. Dominant allele, an allele that expresses its effect even when present in a single copy, heterozygous condition, masking the effect of a recessive allele, represented by a capital letter. Recessive allele, an allele that only expresses its effect when present in 2 copies, homozygous condition, and is masked by a dominant allele in heterozygous individuals, represented by a lowercase letter or small letter. Homozygous condition, a genetic state where an individual has 2 identical alleles for a particular gene, double dominant or double recessive. Heterozygous condition, a genetic state where an individual has 2 different alleles for a particular gene. Capital T with small t, homozygous dominant condition. Individual has 2 copies of the dominant allele, capital T, capital T. Heterozygous dominant condition. An individual has 1 dominant and 1 recessive allele, but the dominant trait is expressed. Capital T with small t, homozygous recessive condition. An individual has two copies of the recessive allele, small t and small t, allowing the recessive trait to be expressed. Genotype, the genetic makeup of an organism, represented by the combination of alleles it possesses for a specific gene. Phenotype, the observable characteristics of an organism resulting from its genotype and environmental influences, e.g., eye color, height, blood type. Across, the mating or breeding of two organisms to study the inheritance of specific traits. Selfing, the process of self-fertilization, where an organism fertilizes itself, commonly occurring in plants. First filial generation, F1. The first generation of offspring resulting from a cross between two genetically distinct parents. Second filial generation, F2. The generation produced by interbreeding individuals from the F1 generation. Trial questions. Pause and try out the questions. This is the result for the F1 generation. This is the result for F2 generation. Reasons for the study of genetics. Understanding heredity. It explains how traits and diseases are passed from one generation to another. Medical advancements. Helps in diagnosing, treating, and preventing genetic disorders. Agriculture and food production. Enhances crop yield, pest resistance, and animal breeding. Evolution and biodiversity. Provides insight into how species evolve and adapt. Forensic science. Assists in crime investigations through DNA analysis. Biotechnology and genetic engineering. Enables advancements like gene therapy and GMOs. Personalized medicine. Helps develop treatments tailored to an individual's genetic makeup. Conservation biology. Aids in protecting endangered species through genetic diversity studies. As we continue, don't forget to subscribe and share. Characters in humans that are inherited in the Mendelian fashion include albinism. Watch my lessons in albinism, variation, and selection, where we dive into these conditions, their symptoms, inheritance, and management. A test cross in genetics is a cross between an organism with an unknown dominant genotype and a homozygous recessive individual. This is done to determine whether the unknown genotype is homozygous dominant or heterozygous. How it works. If the unknown individual is homozygous dominant, all offspring will show the dominant trait. Suppose a plant has purple flowers, dominant trait, but its genotype is unknown. A test cross is performed by crossing it with a plant that has white flowers, homozygous recessive. If all offspring have purple flowers, the unknown genotype is homozygous dominant. That's the genetic cross. If the unknown individual is heterozygous, the offspring will have a 1-to-1 ratio of dominant and recessive traits. A test cross is useful in determining genotype and studying inheritance patterns. That's the genetic cross. As I told you, this is a comprehensive lesson. Let us dive into sex determination in man. Sex is determined by genes located on the sex-determining chromosomes. The chromosomes that determine sex are X and Y chromosomes. Inheritance of the double X chromosomes results in a female human being, and inheritance of the XY chromosomes results in a male human being. Females produce all their eggs, ova, with X chromosomes, and they are homogametic. Males produce sperms with half X and Y chromosomes being heterogametic. The random fusion of the X or Y sperm-containing chromosome with an ovum-containing chromosome results in half males and half females in a ratio of 1-to-1. Let double X represent female chromosomes. Let XY represent the male chromosomes. Parental phenotypes, male with a female. Parental genotypes, XY with XX. Meiosis leads to the formation of gametes, where chromosomes separate. Random fertilization to get the offspring genotypes. The offspring phenotypes are 2 daughters and 2 sons. 50% chances. Remember, this is probability. It can still fall on the side. Linkage. Genes or alleles are referred to be linked if they are carried on the same chromosome and are transmitted along with the chromosomes. In humans, the most common form of linked genes is carried on the X chromosomes and are called sex-linked genes. Sex-linked genes are transmitted along with the sex chromosomes, but they are not related to the sex of an individual. Sex linkage. This is where genes that don't control sex are located on the sex chromosomes and are inherited together with the sex chromosomes. Sex-linked characters. These are characters controlled by genes located on the sex chromosomes and are inherited together with sex chromosomes. Examples of sex-linked characters: hemophilia, the bleeder's disease, red-green color blindness, premature balding. Characteristics of sex-linked characters. They are usually recessive. They occur on the X chromosome. Most of them are disadvantageous. They occur at a higher frequency in males, i.e., males are either normal or victims, while females are carriers. Normal or victims. Let us dive into hemophilia. Hemophilia is a blood condition associated with the failure of blood to clot. It takes long to clot. It is a fatal condition. It is caused by a recessive gene carried on the X chromosome. Symptoms of hemophilia. Hemophilia is a genetic bleeding disorder characterized by an inability to clot blood effectively. The symptoms include prolonged bleeding. Cuts, injuries, or surgeries result in bleeding that lasts longer than normal. Frequent nosebleeds. Spontaneous or frequent episodes of nosebleeds without apparent cause. Unexplained bruising. Large or deep bruises that form easily even with minor trauma. Joint pain and swelling, caused by internal bleeding into joints, especially the knees, elbows, and ankles. Blood in urine or stool. Indicates bleeding in the urinary or gastrointestinal tract. Excessive bleeding after vaccinations. Prolonged oozing of blood at injection sites. Intracranial bleeding. Rare but life-threatening, leading to headaches, vomiting, or neurological symptoms. Let us try two normal individuals, with a carrier woman, a female carrier, and a normal man. Let capital H represent the allele from formal blood clotting. Let small h represent the allele for hemophilia. Parental phenotypes. Normal man, with a normal woman in the physical appearance. Parental genotypes. A normal man with a carrier woman. Meiosis leads to the formation of gametes. Chromosomes separate along with their alleles, followed by random fertilization to form offspring genotypes. One carrier daughter and one hemophilic son, others all normal. Offspring phenotypes. All daughters are normal. One normal son and one hemophilic son. You can try out the following combinations. Management of hemophilia, replacement therapy. Regular infusion of clotting factors, factor 8 or 9, to replace missing or low clotting factors helps the blood clot properly and prevents spontaneous bleeding. Demopressin, a synthetic hormone that stimulates the release of stored clotting factors in mild cases of hemophilia. It can be administered as an injection or nasal spray. Bleeding medications like emicizumab, to prevent bleeding episodes in people with hemophilia A by mimicking the function of the missing clotting factor. Antifibrinolytics, medications that help prevent clots from breaking down, useful for dental procedures and minor cuts. Physical therapy helps manage joint damage caused by bleeding and improves mobility. Gene therapy, an emerging treatment that aims to introduce a functional copy of the defective gene responsible for hemophilia. First aid for minor cuts, applying pressure and bandages for minor cuts, and using ice packs for small areas of bleeding under the skin. Lifestyle modifications, regular exercise to strengthen muscles and protect joints, avoiding contact sports, and using safer pain medications like acetaminophen. Expression in men. Men only have one X chromosome. If the X chromosome they inherit carries the faulty gene for hemophilia, they develop the disorder because they do not have a second X chromosome to compensate. The Y chromosome does not have a copy of the hemophilia gene, so it cannot mask the effect of the faulty gene. Carrier status in women. Women have two X chromosomes. If one X chromosome carries the faulty gene, the other X chromosome usually has a normal copy of the gene, which can compensate for the defect. These women are called carriers. They typically do not show symptoms of hemophilia because the normal gene on the other X chromosome produces enough clotting factor. Rare cases in women. Hemophilia is rare in women because it would require both of their X chromosomes to carry the faulty gene, which is unlikely unless the father has hemophilia and the mother is a carrier. As we end this lesson, let us talk about two confusing conditions in genetics: codominance and incomplete dominance. Incomplete dominance. This is a condition whereby some genes don't show complete dominance. One gene is neither dominant nor recessive over the other. Thus, the F1 offspring are intermediate between the two parents in appearance. For example, when a red-flowered rose plant is crossed with a white-flowered one, the offspring produces pink flowers, an intermediate color between white and red. The heterozygous condition produces different phenotypes from the homozygous state of either allele. Genes responsible for incomplete dominance are represented by capital letters, but of different kinds. For example, if capital R represents an allele for red petals, then an allele for white petals will be capital W. Examples: a red-flowered plant was crossed with a white-flowered plant. All the F1 generation offspring had pink flowers. Use genetic crossings to show the genotype and phenotype of the F2 generation. The genetic cross will resemble that. Capital letters are used in this situation. The offspring are different from their parents, selfing to get the F2 generation. Red flowers, pink flowers, and white flowers in the ratio of 1 to 2 to 1. Try out this question. Show the genotype and phenotype of their offspring. Codominance. It is a condition where genes controlling contrasting characteristics are neither dominant nor recessive over each other. The F1 offspring do not show intermediate characteristics, but instead, both characteristics of the two pure-line parents express themselves independently, and both characteristics appear in the offspring, both genes or characters or dominant. For example, with a bull with a black cow, the offspring have both white and black patches. The genetic crosses of incomplete dominance are treated like those of incomplete dominance, where the two genes are represented with capital letters of different kinds. Examples include AB blood groups and sickle cell anemia. Watch my lesson on sickle cell. Black bull with a white cow, all the calves are roan, with both white and black patches, F2 generation. Sickle cell is inherited in the codominance fashion. Let us carry out a cross between two normal individuals that give rise to a sickler. This means they are heterozygotes F1. Sickle cell is caused by a codominant allele. Let capital H represent the allele for normal hemoglobin. Let capital S represent the allele for abnormal hemoglobin. Parental phenotypes: normal man with a normal woman. Parental genotypes: carrier man with a carrier woman. A carrier is heterozygous. Meiosis leads to the formation of gametes. Alleles separate to form gametes. This is followed by random fertilization to form the offspring genotype. Such a combination is formed. The first double capital H is normal. The second two are carriers, the last one is a sickler. Offspring phenotypes. The first three are all normal. The last one is a sickler. Watch my video on sickle cell management. I will post scenario questions, subscribe, and hit the notification. When you hit the notification button, you will always get updates. Gain access to our well-illustrated notes, activities of integration, assessments and quizzes, and exciting project work.